Okay, let's go ahead and talk through a couple of these real quick, and then we'll move on, and I think I'll finish today. It'll be nice. Okay, so number 6 focuses in on the idea of riboswitches, right? And we're shown two different structures, one of which, when the flavin is bound, we have a transcription termination loop present, right? A termination signal is there. We see this GC-rich stem loop and a poly-U sequence at the 3' end. In the other structure, where the flavin mononucleotide is not present, then that stem loop, that termination signal, is absent. So the idea here is that when there's a high amount of flavin around, transcription of these genes don't occur because this termination signal is present. So we don't get transcription of our structural genes. Therefore, they're not made. When the concentration of the flavin mononucleotide is low, then the termination stem loop is not there. Structural genes get expressed. And as these structural genes get expressed, it leads to the synthesis of riboflavin. Okay? Number 7, it's the idea of attenuation and the fact that attenuation is prokaryotic specific. Remember, this mechanism relies on the ribosome actively translating using mRNA that's actively being made, right? So as RNA polymerase is making the mRNA, the ribosome is bound to that chain as it's being transcribed. This cannot happen in eukaryotes because transcription and translation don't happen in the same compartments. Transcription and replication happen in the nucleus where the DNA is, and translation does not happen in the nucleus. Number 8, so this is kind of applying your knowledge type deal, right? So in this image, what you're shown is a 5' leader sequence where there's no structure shown. You're shown the coding region where this is where like all of the structural genes might be found. And then at the 3' end, this would be called the 3' leader, we see a bunch of different structures present in our RNA. These are called iron response elements, and we'll talk about these later in the semester. But the idea here is that when there's structured RNA at the 3' end of the mRNA, it really can't impact transcription or translation in the same way as it can at the 5' end, right? The 5' end is where we can think about exposing or hiding the ribosome binding site. So just like we saw with one of the riboswitches, I can't remember which, in the presence of an inducing molecule, the ribosome binding site became exposed, right? When the ribosome binding site is exposed, translation can start. Without the ribosome binding site present, translation has no chance of happening. Okay? That can't happen if the structured elements are at the 3' end. We also can't control transcription at the 3' end, right? So the idea with transcription is that we are exposing or hiding a termination signal for transcription. So it'd be complete waste to terminate transcription after all of the structural genes are already made, right? That would make no sense in terms of regulation of transcription. So both of those, translational and transcriptional regulation, need to happen at the 5' end of the mRNA, which means these 3' elements can't really be important in controlling things in that way. So you can kind of think about other ways that that might happen. So perhaps these elements are recognized by some protein that leads to degradation of the mRNA, right? So, or maybe it stabilizes the mRNA. And that's really what 3' elements boil down to, is if there's a protein that recognizes these 3' elements, then it'll bind to them, and that very commonly stabilizes the mRNA, and it will actually lead to increased translation because the mRNA sticks around for longer. So each mRNA that gets made can have a larger impact because it's stable for longer. It doesn't get degraded as quickly. Okay? Any questions? Okay, so then that brings us to thinking about some themes from eukaryotes. Okay, some of the things we talked about for prokaryotes directly translate into understanding things for eukaryotes. But eukaryotes are inherently more complicated, so not all of them are going to directly apply, right? This is just, I would say, a simple image, but it's really not simple, right? But it represents the idea that where the DNA is, where transcription regulation has to happen, is completely separated from where lots of other things happen in the cell, right? So if there's an extracellular signal, that signal has to be recognized outside the cell, translated inside the cell. And in Biochem 1, y'all talked about a couple of these different mechanisms. I know for sure you talked about G protein coupled receptors, right? G protein coupled receptors could initiate this signal relay system. I think you also talked about the the insulin receptor, right? One of our tyrosine kinase receptors. Those are both types of receptors. There's lots of other ones. But the idea here is there's an extracellular signal that gets recognized. It impacts something inside the cell, which initiates a relay. That relay gets propagated through a bunch of different proteins, quite complicated, and unless like you are specifically studying signal transduction, then there's just no point in knowing all this crap, right? But the idea is they're passed through a bunch of different proteins and ultimately these signals are all passed through into one of the core transcription factors that are found in the nucleus. Okay? There are themes here that we're not going to take time to really explore deeply. But the big idea is that we have to have more sophisticated systems because there's just a more complicated system. Okay? In addition to compartmentalization, the structure of DNA is more complex, which means regulation of transcription is inherently more complex, right? In prokaryotes, the DNA is floating around, right? It has some structure. It gets compacted through supercoiling, but nothing like we see with eukaryotes. With eukaryotes, we have these long chains, right? It's packed into chromatin, built of histones. There's just a lot of things going on. So it's packaged differently. There's a lot more DNA to deal with. And the organization of the genome is just different and more complex. So there's a lot more things to think about. And I highlight here the fact that the genomes are less accessible. And that's important, right? Because in prokaryotes, we can say there's one protein that can bind to any piece of DNA it wants to. Because it's pretty true, right? The DNA is generally exposed. Proteins can find it and bind to it. That is just not true in eukaryotes. These proteins have to be in the nucleus to start with. And they also have to deal with the fact that the DNA is packaged in a way that makes it less accessible. It's protected, as it should be. So there has to be ways around this. Gene expression is tissue specific. I really like this table. So this kind of shows if we think about the pancreas versus the liver versus stem cells. Okay, each of these types of cells have very different function. Anybody know the main role of the pancreas? Okay, so right, so insulin, glucagon. So one of the main roles is it secretes hormones. Okay, it also is responsible for secreting a lot of our digestive enzymes. Okay, so we see here that the most expressed genes in the pancreas is carboxypeptidase A1. This is a protein that's responsible for degrading peptide backbones. Trypsin, chymotrypsin, trypsin, elastase, right? All a bunch of genes that y'all talked about last semester that ultimately degrade proteins, right? You talked about how trypsin works. It's the stereotypical serine protease. So because the pancreas is so involved in degrading proteins for digestion, it makes sense that these are the most abundantly expressed genes in the pancreas. Whereas the liver, its main role, and we're going to talk about this a lot this semester, the liver is very involved in metabolism. Okay, so the most expressed genes in the pancreas deal with mobilization of fatty acids. Albumin is a protein in your blood that will bind to fats, will bind to steroids, will bind to cholesterol and move it around. These apolipoproteins serve really similar roles. Here we start getting to ATPase and cytochrome deoxidases. These are really, really important in energy production. Okay, so the liver focuses on metabolism, as is suggested by the overall gene expression profile. Stem cells, right? So stem cells need to be able to differentiate. They need to be able to turn into something else. So they have none of these genes are expressed in an overabundance because they really haven't specialized yet. They're focused on living, surviving, and then figuring out what they need to turn into. So it's a very different, much more balanced expression profile. The most expressed gene here is GAP, which is one of the glycolytic enzymes. Okay. So let's talk about chromatin. What were them I's for? So let's talk about chromatin, right? So chromatin is built up of histone. Like the idea is that it compacts DNA, okay? And at its core, there are repeating units of about 200 base pairs of DNA, okay? And this DNA is wrapped around this histone protein core. The histone protein core is made up of 2 copies of each of these 4 histone proteins, H2A, H2B, H3, H4. I don't care if you know the numbering. I want you to understand that it's basically an octamer of histone proteins that make up a protein core. And the idea is that the DNA wraps around it, okay? So about 200 base pairs wrap around this octameric protein core twice, okay? So here we're looking at our protein core. We see the DNA wrapped around. It makes a full circle. If we look at it from the side, we see that it's actually 2 full wraps. And this image kind of represents it, right? So the DNA wrapping around the outside, the protein core around the inside. And these sequences are linked together by like linker DNA, and that linker DNA is stabilized and protected by this protein H1, another histone protein. Okay, so the idea is the DNA is protected. All right, so the transcription factors, some important themes, is that most of them don't impact DNA at the DNA level. Most of them act from a distance, okay? There can be multiple transcription factors all active at the same time, and most of them will, how ultimately they function is they chemically modify chromatin structure. Okay, this can happen in like lots of different ways. 2 examples are methylating DNA or acetylating DNA. Acetylating side chains of the histone proteins. Methylation of DNA, as we'll see, usually happens on a cytosine. Lysine side chains are ripe for acetylation. They're often pointed outwards and accessible for function for modification. Okay, so here is a methyl cytosine. And so this is the position where deoxyribose would be. Here is our base. Note here, this right here, this is the face where we would have hydrogen bonding. So this is our Watson and Crick face. I point that out because this over here, the position of this methylation, this is where the major groove would be. Okay, so this methylation is functionally modifying the major groove. So you can imagine if regulatory proteins often look for major groove, impacting the major groove might impact how these regulatory proteins are able to bind to it. Okay? It typically inhibits gene expression. Okay? And they're usually found in what are called CPG islands, right? So it's just a cytosine, phosphodiester backbone, guanosine. They're usually these CPG islands, which a bunch of them are present. Most genes are centered around these CPG islands. So they're usually rich in these CPG islands, which makes them very susceptible to regulation. Okay? We'll leave that alone. So with prokaryotes, we talked about usually the transcription factors, like DNA binding domain, is often a helix turn helix. And there's often another structural domain that recognizes whatever the inducing molecule might be. That's also true in eukaryotic transcription factors. We have our DNA binding domain, which typically binds directly to the DNA, and an activation domain, which is going to interact either indirectly or directly with the polymerase. Right? So it's going to impact the ability of RNA polymerase to function, oftentimes impacting the ability of RNA polymerase to even bind. Okay? In terms of the DNA binding domain, there are three major types. Okay? There's something called a homeodomain unit, which is really, really similar to the helix turn helix. Okay? It's not necessarily unique in that sense. These basic leucine zippers are kind of cool. We'll see an image here shortly, but basically it's two long alpha helices that zip together and then at the end kind of open up and bind to the major groove of DNA. And then another one is called zinc fingers. These are called zinc fingers because they rely on zinc to be present, and when zinc is bound to them, they adopt a structure that can bind to the major groove of DNA. So here's the homeodomain. You can see that it looks an awful lot like a helix turn helix. We have a helix, we have a turn, we have a helix. That second helix is interacting in the major groove. Also note here that it is binding to a symmetric part of DNA. So there's symmetry in eukaryotic transcription factors as well. Here's our basic leucine zipper. I mentioned it's two long helices that kind of coil around each other and open up at the end. And this basic region is important because it's interacting with DNA, and it inserts itself into the major groove and makes specific primary contacts. Okay, and the zinc fingers are a little bit different in that usually it's not just one of them that bind to DNA, it's usually three or four. When zinc binds, it forces these sequences to adopt a structure where they can insert themselves into the major groove of DNA. And they actually usually wrap around DNA by interacting with the major groove. So they have multiple points of contact. Any questions before we move on? Okay. All right. So we're reaching the end here. Activation domains. So we talked a couple minutes ago about how a lot of these transcription factors don't often make direct contact with the RNA polymerase, but usually they impact RNA polymerase indirectly. So here we're seeing an example where we have the transcription factor that binds to some sequence divergent from where RNA polymerase binds. And it interacts through this mediator protein. Okay, and I stress here that this mediator is usually a complex of multiple proteins. Okay, they bridge RNA polymerase on the transcription factors. They're redundant and modular. Okay, just meaning that they can have lots of different functions and they can have redundant functions. And often they're very, very synergistic, right? So if one of them is serving one function, another one is serving a complementary function. Okay, and you can have ones that act as repressors or activators, just like with prokaryotic transcription factors. So an example, the nuclear hormone receptors is a very large class that is kind of like a simplified example here because these proteins are inherently, or it's not proteins, these signals are inherently hydrophobic, so they're able to spontaneously diffuse across membranes. So we don't have to have really complicated signaling pathways. These things, when they're made, they're able to get into the nucleus and specifically interact with a receptor. So the idea is that they contain a DNA binding domain, usually a zinc finger domain, and a ligand binding domain that binds to our ligand. Okay, nothing especially tricky here. The ligand being our nuclear hormone. Okay, so the way this works, and this is kind of cool how this works, is So this is our ligand binding domain. We're focusing on this purple helix here, okay? So when estrogen binds, it binds in this binding pocket right here, and that causes this purple helix to change structure. This purple helix actually flips up and interacts more closely with the core of this protein now. And that modification of the structure of this helix 12 is critical in how this all works. Okay, so that modification of this alpha helix is ultimately what leads to the recruitment of the coactivator. Okay, our coactivator is recruited, right? So here in this image here, we're representing how this works. The estrogen binds. When it does, this alpha helix flips in. When this alpha helix is flipped in, it allows the coactivator to come in and bind and interact more tightly. So estrogen binding to our nuclear hormone receptor is what triggers the recruitment of the coactivator. And once the coactivator is recruited, we have a situation here where other things can happen. So this coactivator will recruit, one of the functions of this coactivator is that it will modify lysine side chains. So it takes acetic acid and it puts it onto the lysine side chains. Once these lysine side chains are acetylated, then that recruits this other protein called a remodeling engine. This remodeling engine, as the name suggests, it remodels the structure of chromatin. Right? So what we're trying to show here, like in blue, is a really critical site that's important for RNA polymerase binding. This remodeling engine responds specifically to the modified lysine side chains, which triggers a structural change, exposing the RNA polymerase binding site, allowing transcription to then begin. Okay, so in many ways, there are themes that we saw with prokaryotes, in that ultimately we are responding to some signal, that signal being estrogen, and that leads to the exposure of the RNA polymerase binding site. It's just not as simple. There are other steps in the middle. In this case, the first step is the recruitment of the coactivator because that helix flips in. Once the coactivator is there, part of the coactivator's functional domains will modify lysine side chains of the histone proteins. The modified lysine side chains lead to the recruitment of a remodeling engine, which can then adjust the structure of chromatin, exposing the RNA polymerase binding site, which will then let transcription start. Right? So the same overall goal of allowing RNA polymerase to start, to bind, to promote transcription, but other steps are involved. Okay? And